Hello, everybody. This is Chapter 4, Membrane Structure and Function, and these are your lecture notes. So what does the plasma membrane do? It separates the internal environment of the cell from its external environment. It regulates the entrance and exit of molecules in and out of the cell, and is also responsible for homeostasis so that it is a steady internal environment. So the plasma membrane is a phospholipid bilayer with embedded proteins. So it has hydrophilic or water-loving polar heads that face both the inside and the outside of the cell. It also has hydrophobic or water-fearing nonpolar tails. They face each other interior of the membrane away from water. And steroids such as cholesterol in animal cells stiffen and strengthen the membrane. Membrane proteins may be throughout of the phospholipid bilayer, and there are peripheral proteins associated with only one side of the membrane, and integral proteins that span the membrane and connect both sides. And all these proteins are able to move around laterally, and so we have a fluid mosaic model, a pattern of phospholipids, steroids, and proteins in the membrane. So both phospholipids and proteins can have attached carbohydrate chains. So these are glycolipids, or lipids attached to carbohydrates, and glycoproteins, which are proteins that have carbohydrates attached to them. So here is a model of the bilipid layer. We see the heads of the phospholipids and the proteins that are interspersed between all these bilipid layers. So what are the functions of membrane proteins? We have channel proteins, all right, so things can move across the membrane, and they may have a gate that must be open in response to a signal. And we also have carrier proteins that allow the passage of a solute by combining it with and helping it move across the membrane. We also have cell recognition proteins, which are glycoproteins, and these proteins help the body recognize when it's being invaded by pathogens. And we also have receptor proteins and have a shape that allows a specific molecule to bind. The binding causes the receptor to change shape to initiate a cellular response. We also have enzymatic proteins that carry out metabolic reactions directly. The example given here is proteins of the electron transport chain, which carry out the final steps of aerobic respiration. Here is an example of a channel protein that allows the chloride ion to flow to and from. When this is mutated, this causes cystic fibrosis and causes phlegm to be built up in the lungs. So we have carrier and cell recognition proteins. So here we have the GLUT carriers that transfer glucose in and out of the cell. And we have the MHC protein, or major histocompatibility complex, that helps people recognize self from foreign. We have receptor and enzymatic proteins. Here on the right, we have adenylene cyclase, which is involved in ATP metabolism. And so the plasma membrane can regulate the passage of molecules into and out of the cell because it's selectively permeable. Which molecules can freely cross the membrane and which may require carrier proteins and our energy depends on the size and the nature of the molecule, its polarity and charge. So small uncharged molecules can freely cross the membrane. Examples are carbon dioxide, oxygen, glycerol, and alcohol. And they slip in between the hydrophobic heads and pass through the hydrophobic tails. And this is driven by the concentration gradient. So what is this concentration gradient? It means more of a substance on one side of the membrane than the other. And going down a concentration gradient from an area of higher to lower concentration, or going up a concentration gradient from an area of lower to higher concentration, which requires input of energy. Water that is polar would not be expected to readily cross the membrane, so we have special channels called aquaporins, aqua meaning water, pore meaning pore or hole, that enable water to cross the membrane, and aquaporins are present in the majority of cells. Large molecules, ions, and charged molecules are unable to freely cross the membrane, but can cross via channel proteins, which forms a hole or a pore through the membrane. Carrier proteins are specific for the substance they transport, and vesicle formation via either endo or exocytosis. So here is a diagram demonstrating the permeability of the plasma membrane and the way things can travel in or out. Charged molecules and ions are repelled and have to go through channels. Water goes through the aquaporin, and large proteins must be transported through other proteins. So here's a table about the passage of molecules in and out of the cell, and when energy is not required and when energy is required. So energy is not required when it undergoes diffusion or facilitated transport down to a lower concentration, but energy is required for active transport going up the concentration gradient or via exocytosis or endocytosis. So diffusion is simply movement of molecules of an area of higher concentration down to lower concentration, and it'll occur when equilibrium is reached. For example, when a crystal of dye is placed in water, the dye and the water molecules move about until equilibrium occurs. You could also think about spraying on perfume or cologne. It's very strong on you, but it slowly disperses through the room so someone else could smell it, but it's not as strong 10 feet away as it is on you. And solutions contain both the solute, the solid, and the solvent, the liquid. And for most solutions, the solvent is basically water, but there are other circumstances as well. So here is the example of diffusion and osmosis. You place a crystal dye, a little chunkier of solid, it starts to dissolve, there's an area of concentration, but eventually it diffuses out and the whole fishbowl becomes a light purple color. So gases can diffuse through a membrane, and oxygen and carbon dioxide enter and exit this way. So several factors influence the rate of diffusion. Temperature, so as temperature increases, the rate of diffusion increases. Pressure, electric currents, and molecular size. Osmosis, diffusion of water across a differentially permeable membrane. Remember, diffusion always occurs from higher to lower concentration, and osmotic pressure is the pressure that develops in a system due to osmosis. The greater the possible osmotic pressure, the more likely is water will diffuse in that direction. So always remember that water diffuses down its concentration gradient toward higher solute concentration across a selectively permeable membrane. So an isotonic solution is the solute concentration is equal inside and outside of a cell. A hypotonic solution is a solution that has a lower solute concentration than the inside of a cell, and a hypertonic solution is a solution that has a higher solute concentration than the inside of the cell. So here is an example of cells when they're in an isotonic solution. Up here, no net gain or loss of water. They keep their own size and structure. And here's a hypotonic solution. When water enters the cell, it causes the cell to rupture, or with the red blood cell here, causes it to swell. And then a hypertonic solution, when water leaves the cell, which shrivels it and causes it to shrink. So here is an example of the same thing in plant cells. So an isotonic solution, no net gain or loss of water. And a hypotonic solution, the cell gains water and swells a bit, and the vacuole becomes larger, and this causes turgor pressure, which helps keep the plant erect through the cell wall. In a hypertonic solution, cell loses water, and plasmolysis occurs, or some people would pronounce that plasmolysis. Transport by carrier proteins. The plasma membrane impedes the passage of all but a few substances. Substances enter or exit cells because of carrier proteins, and they are specific. So they combine with a molecule or an ion to be transported across the membrane, or change shape to move molecules across the membrane as well. So carrier proteins are required for both facilitated transport and active transport. So facilitated transport explains the passage of molecules such as glucose or amino acids. So neither molecule is lipid soluble, and there is a reversible combination with specific carrier proteins. And like diffusion, ATP is not required because molecules are transported down their concentration gradient. So here is an example of molecules being transported via facilitated transport. So it's just a change in shape, and energy is not required. In active transport, carrier proteins are required, and they're also often called pumps. And the transport of molecules goes against their concentration gradient. And so energy is required, and usually it's in the form of ATP. So once again, they're moving the molecules against the concentration gradient. An example is the sodium-potassium pump is especially important for nerve and muscle cells. So it moves sodium Na plus out of the cell and potassium K plus into the cell. The carrier changes shape after a phosphate donated by ATP attaches, and then again after it detaches. So here is a diagram of the sodium-potassium pump where it transports 3 sodiums versus 2 potassiums, and it can bind them very, very tightly. So here we have sodium binding. We have ATP converted to ADP for the energy. And then a change in shape results and causes the carrier to release 3 sodiums outside the cell. Now the carrier has a shape that allows it to bind 2 potassiums. Phosphate group is released from carrier, and the change in shape results and causes the carrier to release 2 potassium molecules inside the cell. Now for bulk transport, we do this by vesicle formation. So these are macromolecules that are too large to be transported by carrier proteins. Energy is required to form these vesicles, and it's called membrane-assisted transport. When molecules exit the cells, it's called exocytosis. When molecules enter the cell, it's called endocytosis. So exocytosis, the vesicle fuses with the plasma membrane as the secretion occurs, and then that vesicle membrane becomes part of the plasma membrane. So examples of these are pancreatic cells releasing insulin or other enzymes, or anterior pituitary cells releasing growth hormone. So here is a diagram illustrating exocytosis. So here's the vesicle right here. Sorry, I drew a blank there. It comes from the cell, binds to the plasma membrane, fuses, and then releases the molecules to the outside of the cell. And so endocytosis is just the reverse. Cells take in substances by vesicle formation, and part of the plasma membrane basically invaginates to envelope the substances. Then the membrane pinches off to form an intracellular vesicle, and there are 3 types of endocytosis. phagocytosis, pinocytosis, and receptor-mediated endocytosis. So phagocytosis is large particulate matter such as food molecules, viruses, or whole cells. This is very common in amoeba and macrophages. Pinocytosis is liquids and small particles. This is common in blood cells and plant root cells. And receptor-mediated endocytosis is a type of pinocytosis involving PITS-coded receptor proteins for specific molecules. And this is very efficient and very specific. So here is the example of phagocytosis. It's very self-explanatory. Pinocytosis, similar to phagocytosis, but the molecules or soluble bringing into the cell are smaller. And here's receptor-mediated endocytosis. So there are receptor proteins inside the pit and it becomes a coated vesicle. So there are modifications of the cell surfaces. And so it's made of, our extracellular environment is made of large molecules produced by nearby cells. So plants, prokaryotes, and fungi are surrounded by cell walls. Animals have more varied extracellular environments that can change. So in animal cells, we have the extracellular matrix outside of cells and junctions that occur between cells. And both can connect with the cytoskeleton and contribute to cell communication. So the extracellular matrix, or ECM, is a meshwork of proteins and polysaccharides that are closely associated with cells that produce them. And so they have certain proteins that are there. Collagen resists stretching, elastin provides resilience to the ECM, and fibronectin is an adhesive protein that links to integrin, which in turn links it to the cytoskeleton. The extracellular matrix also contains proteoglycans, which are polysaccharides made of amino sugars that attach to proteins in the ECM. And proteoglycans attach to a long centrally placed polysaccharide. And so it resists the compression of the ECM, and it also assists in cell-to-cell signaling. So here's a diagram of the extracellular matrix showing you the actin filaments, the integrins, the elastins, the collagens, and the fibronectins. So there are also junctions between cells. So cell surfaces in certain tissues of animals have junctions. There are adhesion junctions, which connect cytoskeletal filaments of adjacent cells. There are tight junctions, which are proteins that connect plasma membranes of adjacent cells to form impermeable barriers, and there are gap junctions. And these are plasma membrane channels of cells joining, allowing communication and strength. So here are different diagrams of these junctions. So there's the adhesion junction, the tight junction, and the gap junction. Plant cell walls are generally porous. It contains cellulose microfibrils as the main component. Pectins allow the walls to stretch as cells grow, and non-cellulose polysaccharides harden the wall as the cell matures. Pectin is abundant in the middle lamina, and it's a layer of adhesive substances that hold cells together. Plasmodesmata are narrow channels that penetrate the cell wall to connect adjacent cells. Each channel contains a strand of cytoplasm. Cytoplasm strands allow exchange of materials between cells, and only water and small solutes can pass freely, and that cytoplasm connects all the cells within a plant. Thank you all, and have a great day.